packet we just discussed at the beginning of class was all about transcription, right? Right here. We're also going to be talking about translation today. And at the end of today's class, we're going to get a chance to work on some practice stuff and to see how well we can do these processes. So reorienting ourselves, making sure we're back and we understand kind of what we're talking about. Once again, this entire unit is all about DNA and how it does its job. And the way that it does its job is based on this whole idea of the central dogma, right? These foundational principles, foundational processes that allow life to happen. If it weren't for all three of these things, right, none of us would be here. This is how DNA actually works as the blueprint for life. So DNA replication, we've already covered. We had a quiz over it. You had a project over it. We're now moving on. We're talking about how we actually go from DNA to protein, how we do transcription and translation. Oh, no, my clicker. Yeah, what's up?
What's the unit test going to be?
I don't know if we're going to have a unit test over this.
Why?
Because we've usually had a project over this.
Why? We're going to get our quiz grades up.
Probably quiz Bs.
Oh.
I'll talk to you more as we get closer to the end of the unit and go back and get higher grades on them. Yeah.
So are we going to know when the actual unit?
Yeah, I'll keep you informed. We still got 5.3 and 5.4. We're like halfway through. So we're still going. This is kind of a long unit, but it's like really important. So, all right. So different visualization of that whole concept. This was also brought up in the video. We had DNA to RNA to proteins, right? This is the flow of information in biological systems, at least in living things that use DNA as their genetic code, which is just about everything that's not a virus or something. Right, DNA has the code, right? This is the molecule of heredity. This is what has the genes. This is how information, right, genetic information gets passed from generation to generation, from cell to cell. But what we really have in genes are, for the most part, instructions for making proteins, right? And it's proteins that are actually going to be doing stuff in the cell. So this is all about how we actually get there, right? We have our DNA code. We copy it, part of it. We copy like one gene at a time, for example, right, into RNA. And that RNA can actually leave the nucleus, go out into the cytoplasm, go to the ribosome, where it tells ribosomes which kinds of proteins to build, right? Transcription is that first process. Translation is the second process. Quickly, before we get into them in depth, these words sound really similar. They look similar. They both start with trans. It can be easy to confuse transcription and translation. So let's think about what these words actually mean. And we can use language analogies to kind of understand them and make sure we don't get them swapped up in our brains. So I believe there was a question in the packet about this too, but what is a transcript? Well,
Copy.
It's a copy, exactly. It's fine. I love when people answer my questions that I ask. It's great. A transcript is a copy, right? To transcribe something is basically to copy something, right? Once upon a time, there were people known as scribes. And their whole job was to copy things. Generally, it was like a monk sitting in a monastery in France, in 12th century France. And he had a Bible, and he had a beer, and he had paper and ink. And that's the other thing they did. They copied Bibles, and they would take silent vows, and they brew beer. That's like the life of a monk. So sits there. Here's his Bible. Here's a paper. Copy. Letter for letter, word for word. Letter for letter, word for word. Yeah, it was. Before the printing press, books were like really, 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 really expensive and nobody could read. All right, so to make a transcript of something is just to copy it letter for letter, word for word, right? So we're staying in the same language. We're going to use this language analogy, right? DNA and RNA, they don't really have a language, right? We use these letters. These letters aren't actually letters. They're not actually words. They're chemicals, right? These are actually chemical compounds, right? But we can kind of think of it as like the language of DNA is it's a 4-letter alphabet, A's, T's, C's, and G's, right? A, T, G, C, T, T, T. What do we have down here? A, U, G, C, U, U, U. It's the same information. Slightly different dialect because we're using U's instead of T's, but otherwise it's exactly the same code, right? We're staying in the same language. We're simply making a copy, right? That's what transcription is. We're making a transcript, a copy of that information. Translation is what happens across the hall in Japanese class or French class or Spanish class, right? This is where we're changing languages. That's exactly what we're doing here. We're taking this information in the language of nucleic acids, right? Those bases in the nucleotides, A's, C's, T's, G's, and U's. And we are using that information to go into a totally different language, the language of proteins, which are amino acids, methionine, leucine, serine, tyrosine, totally different kind of molecule, right? But we were able to use the information from one language to write something in a new language. And that's exactly what you do in Spanish class, right? You're translating. So here, we're simply making a copy. We're staying in the same language. We're still using A's, T's, G's, and C's, except U's instead of T's, right? Here, we're changing to a totally different language, totally different type of molecule. So hopefully that can help you keep straight transcription versus translation. All right, big idea. We're just looking at the whole big picture here. Why proteins? Why is this so important? Why does DNA have to tell proteins what to do or what kind of proteins to build? Well, proteins are basically the molecules in the cell that make it work. If it weren't for proteins, nothing would get done. All those enzymes we talked about that help like copy DNA or make RNA copies, helicase, primase, polymerase, ligase, all those ASEs, all those enzymes are proteins. They're one class of proteins are called enzymes, and they do all kinds of important stuff. You can kind of think of the DNA, right? We've talked about being like the control center or like it's in charge of the cell. You can kind of think of that as being like the CEO of a company, right? And since we're using blueprints, we can make it a construction company, right? So the CEO is like, the DNA is kind of like the CEO of a construction company. But the CEO of a construction company doesn't actually pour the concrete or frame up the walls of the house, right? That's what the workers do. So DNA is kind of like the CEO. mRNA are kind of like the managers, or maybe like the foreman on the job, right? But then the proteins are the actual workers. They're the construction workers who are actually getting in there, getting dirty, and doing the work. Proteins do things like build cellular structures, control biochemical pathways, move stuff around in the cell, regulate various processes. If there's something that needs to get done in a cell, there's probably a protein involved in it, right? Like, for example, active transport of something across the membrane, or opening up to allow facilitated diffusion of glucose into the membrane, right? Those are a couple of different proteins we talked about, right? All those different enzymes we talked about. When there's something doing something in the cell, there's almost always a protein involved. Next couple slides are running through some vocab terms. I'm going to go through them kind of quickly, but you can always come back to them later. I think I got a Quizlet on Canvas, and if not, you can come back to this and make a Quizlet yourself if you want to. Protein is simply a macromolecule. It's made up of amino acids, and they do all those different things that I was just talking about. They're also known as polypeptides. Once again, as a reminder, when you see polypeptide, just think protein. They both start with P. Hopefully, it's pretty easy to keep straight. Proteins are polymers, right? Which means that they're made of monomers, and the monomers of proteins are amino acids. So this is our fourth and final major class of biological macromolecules we're talking about during freshman biology class. If you remember last semester, we talked about lipids, we talked about carbohydrates or sugars, and now this semester we've talked about nucleic acids, DNA and RNA, and now we're talking about proteins, right? So proteins are our last one that we're talking about, and they are made out of amino acids. And these are linked together chemically. We just chain them together in a sequence to make bigger proteins, and they're sometimes called the building blocks of life, kind of like how atoms are the building blocks of matter. All right, so essentially, there are a bunch of different types of amino acids. We put them all together in a big old chain. That's going to fold up into three-dimensional structures that are going to have particular functions. Cool. Any questions there about protein versus amino acid, polymer versus monomer? Boys, are you with me? Okay. All right. A few other vocabulary terms. These are things that are going to pop up a lot. First of all, what is a gene? A gene is simply a sequence of DNA, right? A stretch of those A's, T's, C's, and G's that code for something, usually a protein. Not always, but usually. Ribosomes are the little cellular machines that build proteins. You guys learned about that back when we had our first unit, right? You built cell models. Remember that? Oh, man. Good old days. A codon. Codon, this is something that's going to be coming up. Once again, think about our language analogy, how we have our little four-letter alphabet for DNA and RNA. A codon is essentially a little three-letter word that DNA and RNA use. DNA and RNA can only make three-letter words. They never got above a hop-on-pop reading level. Yeah, Dr. Seuss. Okay, so that's what a codon is. We'll come back to that concept here in a little bit. Transcription, that's making RNA from DNA. Like I said, right, we're making a copy. Translation is where we take that information, the mRNA, and we build a specific protein-based information, right? Changing languages from nucleic acids to proteins. Any questions about any of those vocab terms? Once again, you can come back to if you need to. Yeah.
Are we gonna have to use the codon chart?
Mm-hmm. I've got some up here for you. I'm going to pass them out to you later.
I like them.
Yeah, it's fun.
Pretty easy.
It's fun.
And it makes you feel smart.
Good.
You just go like that.
See, look, something to look forward to later today. All right. Okay, a little bit of information about amino acids forming a protein. Basically, when we chain together a bunch of amino acids, we get a protein. There are 20 different amino acids that organisms on this planet use. Now, there could be lots of other ones. And in fact, in laboratories, we've synthesized all kinds of invented amino acids. All you need is an amine group on one end and a carboxylic acid group on the other end. That makes an amino acid. You could have anything in the middle. All right. But these are the ones that, as far as we can tell, all of the organisms from the lowliest bacteria to you, all right, use to build our bodies. There are 20 of them. And depending on what order you put them in, you get different proteins, right? Make sure you memorize the structure of all 20 of those. That's a joke. Yeah, I am a joke. I'm joking. Yeah. I don't have. Did I say I'm a joke? I'm a joke. All right. I've never done that. You shouldn't. Okay. Maybe like if you're like getting your master's degree in biochemistry, you might memorize all 20 of them. Maybe, but like you're not. You're a freshman in high school. Please don't memorize all 20 of those.
Don't worry.
I was just making sure you're awake, right? I think I was told to memorize some of them, maybe all of them, when I was in a biochemistry class in college, and I was like, guess I'm getting a B on this test. So
That's how I feel.
you don't have to memorize those. All right, so proteins, right? Once again, they are macromolecules. They're made up of amino acids. Those are the monomers. Some of them might be really short, might just be dozens of amino acids long. Some of them are really, really huge, thousands of amino acids long. You got big proteins, you got small proteins. All depends on what they're going to be doing. And once again, it's the specific sequence of amino acids that determine the shape of the protein. Oh, yeah, real quick. If we go back and look at those amino acids, right, they're all different. Some of them are big, some of them are small. Some of them are positively charged, some of them are negatively charged. Some of them are neutrally charged. They don't charge at all. Some of them are hydrophilic, some of them are hydrophobic, some of them are polar, some of them are nonpolar. They all have different chemical properties. And because of those chemical properties, when you have different amino acids in different sequence, they're going to fold up on each other differently into different three-dimensional shapes, right? So that three-dimensional shape that a protein is, though, this is what determines its job. So hopefully this sounds familiar to all of you. Some people earlier today told me they'd never heard it, which is just not, it's not true because I said it earlier this semester, and I said it many times last semester. If you didn't hear from your other biology teachers last semester, blame them. Form follows function. Good. At least a few of us have heard it. Yeah, FFF, form follows function. It's alliterative. It's a core principle idea in biology. The shape that something is that shape in biology. It's evolved to be that shape because it has a particular job that it has to do. If you change the shape, you will change the function. You have to leave, right? Yeah. Adios. Have a great day. Do you want the practice sheet so you can do a little practice? Yeah. All right.
Thank you.
You are welcome. All right. So the shape of the protein determines its function. If you were to change the shape, it would no longer be able to do its job properly. This protein here is hemoglobin. Anybody know what hemoglobin does?
Blood carries oxygen.
Yes, carries oxygen in your blood. Madison, I'm sure you knew it as well. The way that it does that is if you see these little pockets, this is actually a very big protein. It's actually 4 separate proteins all shoved together. That's what the different colors mean. Each of these little pockets you see here is just the right shape to be able to hold a compound called heme. And that heme compound contains iron. And the iron in the heme can bind to oxygen and hold on to the oxygen, right? And that's what allows your blood to pick up oxygen in your lungs and then carry it and transport it to your cells, your tissues, all throughout your body, right? If this pocket was a different shape and it was not able to hold on to heme, then your red blood cells would not be able to carry oxygen. And you would be very, very, very dead, okay? We try to avoid being dead here in biology class if we can. All right, so form follows function. The shape of the protein determines what kind of abilities that protein is going to be able to have. Quick review over DNA and RNA. DNA double-stranded, RNA single-stranded. DNA uses deoxyribonucleic sugar, RNA uses ribose. And of course, DNA uses CGs, A's, and T's. RNA uses CGA and U. We've already covered this before. If there are any questions about that one, we all know that stuff, right? Good. All right. So when we're going to make RNA, this whole process of transcription that our packet was over earlier, okay, quick rundown of that stuff. It's done by this enzyme here, RNA polymerase. Just like how DNA polymerase built the DNA polymer, RNA polymerase is building our RNA polymer. And it does so by reading one strand of DNA in order to make a copy of the other strand, right? So if you look at this, we're using our base pairing rules as we move along, reading from 3' to 5', building from 5' to 3', just like we talked about before, same as DNA polymerase, in order to make an exact copy of the non-template strand. You can see that all those bases match up there, except of course we're using uracil instead of thymine because it is RNA. So we read the template strand to make a copy of the non-template strand. Any questions there about the basics? Yeah.
Does this happen after, during, or before replication?
It will happen at the other times. So when DNA is not being replicated during the S phase, like during the G1 and the G2, that's when this will be happening.
Does it ever just sit still?
Still, you die.
Like the DNA, does it ever just like?
Well, certain segments of it are off, and the genes are off, and they don't get transcribed.
It just seems like it's always doing.
Yeah, your body is just doing all the time, just doing stuff all the time. But it's good because otherwise, dead, very dead, dead. You got to keep running just to stay in the same place, man. Welcome to the rat race. All right. RNA transcription. So if I were to give you a DNA code and told you to write down what the RNA code would be, how would we do it? If we read a G, we would put a
C.
If we read a T, we'd put a
U.
A.
A.
If we read an A, though, we put a
U.
U. C, you put a, right? Yep.
U.
All right. Just like
I
that.
wish I could do it that fast.
What?
I wish I could do it that fast.
Right, that'd be amazing.
Mom?
Okay, same idea here. Okay. That's how we do transcription. If I were to give you code, like I'm going to in a little bit. Right, that's how you would transcribe. All right. Any questions about that? Base pairing rules. Don't forget, don't use T's, use U's. Sick. All right. Quick rundown of the different steps of transcription. So we do have 3 stages that are identified based on what's happening when. So you might wonder, okay, how in the world do we know to just make a copy of this one gene? How does RNA polymerase know to just copy this bit and not copy the entire dang thing? The way that it does that is by recognizing certain sequences in the DNA that are known as the promoter and the terminator sequence, okay? So the promoter promotes transcription, right? Encourages. Think of it that way as in promoting. So it's a sequence of DNA bases that the RNA polymerase enzyme will recognize and will bind onto and then start unwinding and reading the DNA strand. So that whole process, when that happens, is known as initiation because we were initializing or beginning. Then we have elongation. This is where our RNA polymerase enzyme is going to move and it's going to read the template strand from 3 to 5' and build mRNA from 5' to 3', right? Because we're always doing that anti-parallel stuff. And it's going to do that using free-floating RNA nucleotides that are in the nucleus, just chaining together those monomers to make the polymer. Eventually, though, it'll reach another signal, another sequence of bases in the DNA that tell the RNA polymerase, hey, this is the end of the gene. You can stop now. This is the terminator. And the RNA polymerase is like, all right, bye, but I'll be back.
Okay.
Nobody's ever seen Terminator
No, Arnold said
or knows Arnold Schwarzenegger because of the Terminator, right?
He's good. Yeah.
It's okay, you don't have to laugh. All right. The termination of the signal, basically, it's the end of the gene. It's the terminator. I'll be back. Is a robot from the future. It's the whole thing. All right.
That was humorous.
Yeah, thank you. All right. Any questions there about those 3 different stages of how we do transcription? Yes, Keegan. That's not a question I'm answering. I don't know.
Yeah, 7.
Which one are we on?
Should I just let you practice without even knowing how to do it yet? Is that what you should do?
I'm back. I know how to do it, dog.
Oh.
I can.
That's true. Those of you who are in biomed are like, I already learned this 2 weeks ago.
About halfway.
All right, we're more than halfway. mRNA processing, okay? We already learned about this. It's where we add the cap and the tail. We cut out the introns, leave the exons. Any questions about that? Yeah?
Can we take a half time?
13. All for the form.
We'll get up again here in a little bit. All right. These are just a couple of images showing the mRNA editing. Once again, cutting out the introns. We already covered it earlier in the packet. Any questions about it? We're good? Good. All right. So moving on, though, we are going to be talking now about translation. But before we talk about exactly what happens in translation, we need to talk about two other types of RNA. So the RNA we've been talking about building during transcription, that's mRNA, messenger RNA, and you've already learned about it. This is what we've been discussing. This is what our messages are transcribed as. It's messenger RNA, right? This is what contains the codons, or the little genetic words that signal to the ribosome to use particular amino acids. That's mRNA. Are there any questions about the basics of mRNA? Single-stranded, has codons. It's a copy of the DNA, right? It's a transcript. Cool. Those codons then are read by the ribosome, and the ribosome will be able to make the right protein. But in order for the ribosome to do its job, there are two other types of RNA that are involved, which we'll get to after I forgot there was a codon slide. Sorry. Codons, three-letter words, little three-letter genetic words, right? For example, AUG, that's a codon. ACG, that's a codon. GAG, that's a codon. Right, once again, three-letter words. It's as long of a word as you can make using DNA or an RNA. Every one of those codons, every one of those little signals of 3 nucleotide bases are going to code for a specific amino acid, or they will code for stop, which will tell the ribosome to stop building the protein. All right, now we get back to tRNA and rRNA. All right, next up RNA is transfer RNA. You can also think of it as transport RNA if that makes it easier for you. And its job is to carry or transfer the amino acid monomers that we're going to be using to build our protein to the ribosome. So these carry those monomers to the ribosome. The important thing about tRNA is that they also have this region down here that's called an anticodon. And the anticodon, remember anti means opposite. This is a codon essentially that is the opposite bases of an mRNA codon. So they'll be complementary. So for example, the codon is CAC. The anticodon would be GUG. That one would match it, right? And these will fit together in a later stage in translation in order to make sure that we get the right amino acid in the right spot of our protein. They have this kind of clover leaf shape, but you can kind of almost think of it like a T if you want to. Kind of looks like a T. T for tRNA. Might help you recognize it. All right, so these are really important because these are what's carrying our amino acids. And then finally, we have ribosomal RNA, or rRNA. And this is what the ribosome itself is actually made out of. The ribosome is made of rRNA, and it has 2 units. There's a large subunit, there's a small subunit, and right in between is where our mRNA will go. Just like you saw in that animation earlier during the video. Okay, and of course, the ribosome is where we actually build our proteins. So in order to make a protein, in order to do our translation step, to finally get that protein that we need all 3 types of RNA. You have to have the rRNA in the ribosome because the ribosome is what is our protein building factory, our little protein making machine. You have to have the instructions from the DNA. Otherwise, you don't know which protein to make. And that's carried by the mRNA. And you need your tRNA because they are what is bringing in the correct amino acid. Without the amino acid monomers, we can't make the protein polymer. So if you're missing any one of those components, you're not going to be able to do translation. So all of our RNA is working together in order to make proteins. Any questions about those 3 different types of RNA before we move on? All right. So you got to see this animated already in the video. So hopefully this is not too foreign looking to you. But once again, this is translation. Here is our mRNA, right? Our message. In the mRNA, we have our codons, right? Those codons are read by the ribosome, which is our orange thing here. And as the ribosome reads, tRNA that have complementary matching anticodons will go inside the ribosome if they match up chemically, right? Just like UUU complements AAA, UCG, AGC, right? Those tRNA that have the complementary anticodons will match up with the codon in the mRNA inside the ribosome. And then the ribosome will pluck off the amino acid that they are carrying, right? Just like this one's missing one, right? They'll pluck that off and add it on to our growing protein chain there. Any questions about that? So that's where all 3 of our types of RNA are being involved. They're all doing their job properly here. This is just the same thing, just a different diagram, slightly more detail, but it's the same idea. It's just kind of upside down. I don't know. I included them both because I don't know. Cause. All right. Polypeptide, why is it called a polypeptide? The reason why proteins are called polypeptides is because we have a special name for the bond between amino acids. They're called peptide bonds, and they're formed using dehydration synthesis. What?
Yay!
You thought you didn't need to know that anymore. Just
That's one that H is
kidding.
out.
Anytime we're doing polymerization, building big molecules, we're going to be doing dehydration synthesis, right? Removing an OH from one, removing an H from the other, pulling out that water to make a new bond. And that's called a peptide bond. That's why they're sometimes called polypeptides. It just means many peptide bonds. Polypocket, sure. All right. So we got an amino. This is the amine group. This is the acid group. Take the OH from the acid on this one, the H from the amine on that one, and we form a new bond. And that whole special thing, that's the peptide bond right there. All right. 20 different amino acids. Okay. Whenever you're doing some of the amino acids, please use the 3-letter abbreviation. And you know you do not need to memorize them. Please do not use the single-letter abbreviation because I don't have those memorized. It's just purely me telling you not to make life difficult on me. Like, I don't know that Q is glutamine. Why would Q be glutamine? I don't know.
G's already
So-
taken.
Exactly.
Isn't
So,
it?
yeah, glycine.
Oh.
Yeah, so please use the 3-letter abbreviation, not the single letter. Cool? Thanks. Appreciate you.
That doesn't even make any sense.
All right. So what you have in front of you right now is what's called a codon table. A codon table is basically a quick and handy tool using the knowledge that biologists, molecular biologists, geneticists, etc., have deciphered about the genetic code. We figured out what every codon means. Isn't that cool? That's kind of amazing, isn't it? Wow, science, knowledge, it's amazing. So this table will allow you to do the translation. If you know an mRNA sequence, then you can figure out exactly what protein it's going to make. And the way you do that is by going these rows here on the left for the first letter of our codon, top ones, the columns for the middle one, and then these over here are for your third position. So for example, GAU, G puts us in the bottom row, A puts us in this column, like, because I'm a PowerPoint wizard, and U, ASP, aspartic acid, right?
What happened to the circle?
Oh,
Can you do
I like
that
the
again?
circle
Yeah, the circle's
better.
better.
Can you do
The
this
circle's
again?
on the next
Harry
slide now.
Potter, though,
That's
right?
crazy.
Wait, no, but like with your hand.
Where's the asparagus?
That was an illusion, or I did it with my hand. It was actually computers the whole time. All right,
There.
I like this one better personally.
That's
Okay,
better. That's
but
better.
you can use
We like that one better.
whichever one you like. Okay, if you like the square one, you can use the square one. If you like the circle one, you can use the circle one. I'm going to give you these in a moment. All right, my recommendation, I hole punched them for you so you can hold on to it and keep it and use it whenever you need it. Okay, you can use the circle one or the square one.
On the
Even
quiz?
during
Circle-
quizzes, right?
Yeah. No, I'm going to make you memorize the whole thing.
I'll find.
How would one use the circle one?
I think.
All right.
it's-
Circle one I think is easier because all you do is you start on the inside and go to the outside. So for example, if we're doing GAU again, G is right here, A is right here, U is right here. Once again, we get ASP, aspartic acid.
It's crazy.
It's easier.
You know how much time it took me to get these shapes to be just the right thing? It was like 6 years ago, but still.
Good job.
Sometimes I'm really proud of my PowerPoints.
I just never imagined we could do something like
It's
that.
because you're a wizard.
All right. So once again, that's ASP. All right. Let's try some more. AGC. Shout it out when you see it.
Sure.
Sir, serine. Good. Yep. Right. AGC serine. CCA.
Pro.
Pro. Yep.
Wait, can someone go slower so I can see if I can do it?
You get a chance here in a little bit. We're going to do some practicing. The last 15 minutes of class. UAG.
Stop.
That's
Stop.
I already knew that
Yeah,
one.
Woo.
What is stop?
All right.
I know that it's stop.
Stop. Stop means stop. All right. Knowing that these mean stop, and they mean, hey, stop building protein. We're done. Right? Which amino acid do you think is going to start every protein?
Oh, AUG.
AUG.
I knew that one.
Which is not the amino acid. What's the amino acid that'll be the beginning of every protein?
Oh, metaphene.
Meth.
MET, which is methionine, okay?
Just
So-
it's meth again.
It's everywhere.
Is it that same meth or is it a different meth?
I don't remember. The ribosome will not begin forming a protein until it reaches start. This is important, okay? Hold on. You're going to get a chance to practice. Whenever you're doing transcription and translation, when you're doing that translation stage, you're reading your mRNA, think like a ribosome. Don't start till you see start. Don't keep going after you read stop. Follow the directions, like street lights, right? AUG methionine is always going to start off every one of our proteins. Every protein that the ribosome builds is always going to start with a methionine amino acid to start that protein. And it'll keep going on and on and on until it reads a stop codon. And there are 3 different stop codons that will tell you to stop. All right. If you have questions or need help, you can ask me questions. But what I've got for you guys, I've got codon tables and I've got
Mr. Hartman,
handouts here.
when someone's being disrespectful, you should say UAG.